My name is Ian Baldwin, and I'm delighted here to be presenting part 2 in a 3-part story on how to study the plant ecological interactions in the genomics era. I'm a director of the Max Planck Institute for Chemical Ecology, in part 2 here, I'll be talking about Nicotiana attenuata, the plant that is right here, ability to be able to respond to attack from a nicotine-tolerant herbivore. I just want to remind you that this is a part 2 of a 3-part series, and in the 3rd part, I'll be talking about the plant's perspective on sex, seeds, and microbes. In part 1, I talked about how the Max Planck Institute for Chemical Ecology came about and how it fits into the rich history of the field of plant-herbivore interactions and how Ernst Stahl in 1888 really started the field. I also talked about the process of training genome-enabled field biologists and how they are trying to phytomorphize themselves and understand what plants are doing with this incredible chemical prowess that they have and how they use those chemicals to solve ecological problems. I also introduced the Ask the Ecosystem approach, which combines both field and laboratory studies of transgenic plants, and introduced the important process of silencing genes to understand their function at a Darwinian level in an organismic context. These field experiments are conducted with these genetically modified plants in their native habitat in a nature preserve in the southwestern deserts of the United States in Utah, in a collaboration with Brigham Young University. What I want to do here in part 2 is talk about this particular interaction that's unfolding to you right here. This is an interaction of the plant that we work on, Nicotiana attenuata, and the hawk moth, Manduca sexta and Manduca quinquemaculata. It's a remarkable interaction filmed here in fast motion, fortunately enough, by the team of Folker Arts from the movie Kluge Pflanzen. They were so kind for letting us use their outtakes. This is a remarkable interaction because the plant is chock-a-block full of one of the most toxic compounds for human beings and for almost any animal with a neuromuscular junction, namely nicotine. Now, many of us have had an addictive relationship with nicotine as smokers, but if any smoker had ever tried to eat a Nicotiana plant, you'd realize just how poisonous this plant is. Nicotine poisons the neuromuscular junction, the acetylcholine receptor called the nicotinergic acetylcholine receptor, and that receptor mediates how muscles move. So if you were a plant and you wanted to design a chemical defense which would poison animals that moved with muscles, this would be an ideal defense compound to produce. And this is exactly what Nicotiana attenuata and some of the other tobacco plants have done. They've evolved this molecule. Now, this molecule evolved from 2 primary metabolic pathways, the NAD pathway and the polyamine pathway, that produce the 2 rings that both contain a nitrogen, and then they're fused together to form the molecule nicotine. Nicotine is synthesized, as I said, from these 2 primary pathways, and its biosynthesis has been worked out by a number of researchers over time. But what is more recent is its understanding of the evolutionary history of this biosynthetic pathway. And this was done recently by Sujing Zhu in our department and a number of his colleagues in the informatics group that is involved in assembling the genome of Nicotiana attenuata, which is currently under review. Now, what Sujing Zhu and colleagues found out was that all of the genes that are involved in nicotine biosynthesis are genes that are part of a whole genome triplication event that happened with the Solanaceae, namely all the plants that are of the group of plants that are called solanaceous plants, potatoes, tomatoes, eggplant, all went through a genome triplication event. Those extra copies of the genes were therefore given the evolutionary privilege to be able to be combined in novel things other than their primary metabolic pathways. And potatoes and tomatoes and tobacco all produce nicotine, but tomatoes and potatoes produce them at much lower levels, about 3 orders of magnitude lower than tobacco plants. Tobacco plants' remarkable ability to produce enormous quantities of nicotine to really make it defensive and smokable has to do with the ability of the plant to have corralled the biosynthesis of those pathways into the roots and to have fused the two rings in a very efficient way and funnel a lot of reduced nitrogen into the biosynthetic pathway. That's described in this paper that is currently under review. Now, nicotine biosynthesis can be inhibited by silencing a single gene. This gene here, putrescine methyltransferase, which we have silenced by RNAi and been able to produce plants that are relatively nicotine-free. And when you make a plant that's relatively nicotine-free and you take it back out into the native habitat and plant it in some natural habitats, you realize just how effective this defense is. Because every deer, every rabbit, every gopher in the neighborhood finds out about it. And here's an example of a gopher that's coming up, has dug a special tunnel up underneath this nicotine-free plant and is pulling it down to its burrows. So without nicotine, the plants become quite defenseless and are stripped bare of their phloem by rabbits and other mammalian browsers, and usually don't last very long. Now, Manduca sexta, which was devouring those plants in that first video that I showed you, is able to do it because it basically holds the world's record for nicotine tolerance. If you compare the LD50s, the lethal dose at which 50% of the experimental population dies, you realize that even the most hardcore Gaulois carton-a-day smoking human being still has an LD50 that is 750 times lower than that of Manduca sexta, which is about 1,500 milligrams per kilogram is able to tolerate. Now, it's been known since the 60s that Manduca sexta's tolerance of nicotine is based on a physiology that allows it to excrete all the nicotine that it ingests without any apparent metabolism or any apparent effect on its nervous system. How it does that is still very much an active area of discovery. But as we look at a caterpillar eating a plant, we've been interested in asking the caterpillar transcriptomically, what is it doing inside of its gut to be able to handle those many human lethal doses of nicotine that it's ingesting almost on an hourly basis. And when you ask the caterpillar transcriptomically, there is consistently one cytochrome P450, which is constantly being regulated in direct proportion to the amount of nicotine that is ingested by the caterpillar. And this is a cytochrome P450 with a long, complicated name called 6B46. And you can see it regulates at a high level when it's eating nicotine-containing plant and down-regulates when it's eating nicotine-free plants. So to understand what this particular gene was doing in the caterpillar and why it was being upregulated every time the caterpillar ate a high nicotine-containing plant. Two scientists in the department, Pavan Kumar and Sagar Pandit, designed a procedure that allowed the study of this particular gene to occur in the natural environment of both the insect and the plant. And what they did was they took that gene, made a double-stranded construct, which is depicted here in yellow in the plant, transformed the plant so that it was consistently expressing this double-stranded piece of the gene that they wanted to silence. And then they planted it out in Utah and let free-ranging caterpillars feed on them. And in that process of feeding on these particular plants, the caterpillar ingests the double-stranded, and then the gene in the caterpillar gets silenced. And in those gene-silenced caterpillars, they were able to understand the function of that particular cytochrome P450, which is upregulated during the defense process. And what they discovered was really remarkable. But let me first show you some data on just how effective this plant-mediated RNA process is. Here on the y-axis is the transcript levels for the particular gene that they're looking at in the various tissues of the caterpillar. And I want you to focus particularly on the midgut, which shows that caterpillars eating nicotine-containing plants have very high levels of that transcript. But if the caterpillars are feeding on a nicotine-free plant, the transcript levels are quite low. But if the caterpillars are feeding on one of these PMRI plants that are expressing a double-stranded construct of that cytochrome P450, and those plants contain normal high levels of nicotine, you would expect the transcript levels to be this high, but instead they're that low. And they're that low because the gene is being silenced by that plant, by the plant's food. And the caterpillars are ingesting that gene, and that RNAi process is happening in basically free-ranging caterpillars in the field. It's a remarkable experimental tool that allows us to study plant-insect interactions in nature using genetic tools to manipulate not just the plant, but also the insects that are feeding on the plant. Now, what's remarkable about this story is that it was actually a wolf spider occurring in the natural habitat of the plant that told us the function of this particular gene in the caterpillar. Now, I'm going to show you a series of videos, and here's a video of a wolf spider attacking a nicotine-free plant, and you can see from that video that it just gobbled it up. So if the caterpillar is feeding on a nicotine-free plant, it has no nicotine in it, and the wolf spider finds this as food. Now, here is, in the next video, is a spider attacking a caterpillar that is fed on one of these PMRI plants. Now, remember, those are full of nicotine, but they are silencing this particular gene in the caterpillar. And you can see from this video that the caterpillar is attacked and eaten as if it was nicotine-free. And this was discovered by the two scientists who had placed caterpillars on plants out in the field, having them feeding on these particular plants that silenced the gene in the caterpillar, and all the caterpillars disappeared at night. And the wolf spider hunts at nighttime, and that's how they found the wolf spider. Now, here is the key moment, the key observation that allowed them to understand what was going on. Because in the next video, here is a spider attacking a nicotine-containing plant that's a normal empty vector wild type plant. And you can see all it did was go up and palpitate the caterpillar, and then it immediately backed away. And what was going on in that palpitation, that little moment when the caterpillar was being assessed by the spider and the spider decided, oh, I'm not going to eat this, was that the caterpillar was through its spiracles. Caterpillars have 17 spiracles. They are basically the lungs of the caterpillar. Caterpillars have all these tubes, and that's how they exchange air. And through the spiracle, the caterpillar is puffing out a load of nicotine into the face of the caterpillar, into the face of the attacking spider. And that's why the attacking spider jumped away. And what this gene is doing is mediating that process in a way that we don't really understand biochemically, allowing the caterpillar to basically divert some portion of that, a massive amount of nicotine that's flowing through its gut that it's excreting out, but then it moves it into the spiracles and it uses it defensively when a spider comes up and says, are you good food? And the caterpillar just puffs out this thing of nicotine and it repels it. Okay? So that shows you that actually, the caterpillar, even though it's excreting most of its nicotine, is using it defensively. It's co-opting just a small fraction of what's going through its gut for its own defensive purposes. But now what I'm going to tell you for the rest of this talk is what happens when the plant recognizes that it's being attacked by that particular nicotine-tolerant caterpillar. Because that recognition process results in 6 changes in the plant that all involve how the plant deals with a caterpillar that has broken through one of its major defenses and has to figure out something else to do with this guy that's going to eat it and is going to make lunch of it. And that recognition process starts right here. And if you look right at that cut leaf edge there, you can see a little bit of green, slimy stuff that the caterpillar is leaving on the edge of the leaf. Now, it turns out it's not doing that intentionally. That's just part of the eating process. It's part of its oral secretions. It's part of the process of masticating the leaves to be able to digest it. But in those oral secretions are a group of compounds that are called fatty acid amino acid conjugates. FACs is what we call them, and the structures of those FACs are right here. They're very simple molecules. They're just fatty acids esterified to amino acids. There's 2 of them. There's 5 fatty acids, and they make basically 8 different structures. And those 8 structures are what the plant uses to say, aha, I'm being attacked by Manduca sexta, and I know that it's nicotine resistant in some way or another. Now, those are anthropomorphizing, but that's basically the message. Now, what I'm going to do, this was, by the way, those fatty acid amino acid conjugates were discovered by Rajko Halecki in his thesis and published back in 2001. What I'm going to do now is to take you through those 6 layers of defense, avoidance, and tolerance that the plant goes through when it recognizes this, that it's being attacked by this particular caterpillar. And those 6 layers are both an up and down regulation of direct defenses, a bunch of indirect defenses, an interaction between indirect and direct defenses, tolerance responses, and avoidance responses. So follow with me, and we're going to go through this remarkable journey of what happens to the plant as it reorganizes its metabolism, physiology, to deal with the fact that it's got a predator that it really has to deal with. Okay, now first I want to talk a little bit about the recognition process. So we've been able to, because we have these synthetic fatty acid amino acid conjugates, we have the elicitors, we're able to start the interaction between plant and its responses without having to have a caterpillar. So we simply just take a pattern wheel and we add these oral secretions, the spit, to the leaves, to the holes that are made in leaves with the pattern wheel, and that elicits a very complicated set of signaling responses. We haven't identified the elicitor, the receptor yet for the elicitor. We know the elicitor, those are the FACs. The receptor is unknown, but that elicits a very complicated signaling network that involves MAP kinases, CYP and WIP kinases, the jasmonate signaling cascade, a lot of modulation of that jasmonate signaling cascade through other kinases, the activation of CDB kinases as well, and the perception by other receptors, LECRK, that it basically involve a regulation of jasmonate signaling. And because caterpillars do not brush their mandibles when they eat a plant, they also contain bacteria and other sorts of bacterial signals, and the plant has to make sure that it's activating a jasmonate signaling cascade and not a salicylate signaling cascade. So all of this signaling has to do with being able to make sure that the caterpillar doesn't fake out the plant with its bacterial signals, but rather generates a nice, clean jasmonate response, which activates 5 out of the 6 layers that I'm now going to talk to you about. Now, that was a lot of work, and that work was done by some remarkable group leaders and remarkable number of talented students that I wish I could talk about in greater detail, but here are their pictures. It also illustrates another important message that I want to bring out in this talk, and that is that interplay between mechanism and function. That if you understand the details by which these responses come about, you have the very tools that you can manipulate genetically to be able to create plants that are not able to show the response. And all of those steps in those signaling pathways have been very useful tools to allow us to be able to manipulate some aspects of these 6 responses in different combinations and test them functionally in the field, in the actual habitat in which the plant evolved. Now, let me go through the 6 responses. The first response was the up and down regulation of these, what we call direct defenses. Now, direct defenses basically can be categorized in 2 groups. They're either toxins, things that poison animals that eat plants without poisoning the plant too much, and are specifically targeted against the things that are different between animals and plants, like nervous systems. Plants don't have a nervous system, so it's really easy for plants to make nervous system poisons that are not toxic to them, but are very toxic to the animals that want to eat them. So in addition to toxins, there's also another type of direct defense that are called digestibility reducers. They're basically interfering with the main reason why a caterpillar wants to eat a plant in the first place, which is to turn caterpillar protein, plant protein into caterpillar protein, to turn plant energy substances like glucose and sucrose and starch into energy substances that the caterpillar can use. So that digestibility process can be interfered with lots of different ways, interfering with all the steps of ingestion and digestion. For example, there are protease inhibitors we're going to talk a little bit about. There are tannins and amylase inhibitors that are basically affecting the digestive enzymes that take apart plant proteins and starches and make them available to be taken up by the guts of caterpillars. But there's also abrasives, things that wear down the mandibles and the teeth of the herbivores, because if an herbivore doesn't have a pair of teeth, a set of mandibles or a set of teeth, it can't chew a plant. And plants fill themselves with silica and other sorts of abrasives that just wear down the teeth. And there is no easier way to starve an ungulate than to wear out its teeth. And plants do that all the time. Now, I just want to talk about the downregulation as well as the upregulation, because the first thing that happens when those FACs are recognized by the plant is that the plant has an ethylene burst and shuts down the very gene that we silence to make a nicotine-free plant. And that's, in fact, the reason why we did it, because we learned from the caterpillar how it was shutting down nicotine biosynthesis in the plant. It's very clear now that since the caterpillar is co-opting a certain portion of the nicotine for its own defense, the plant is most likely downregulating its nicotine production so that the caterpillar can't co-opt the extra nicotine it produces. If it was a deer or a rabbit doing the damage rather than a Manduca sexta larvae, nicotine production would be upregulated 5 or 6-fold, and the plant would become even more full of nicotine than it already is, so that a single leaf would have the same amount of nicotine in it as half a carton of Good Wild cigarettes. So that massive upregulation process is basically stopped, and the plant is downregulated nicotine production when it knows that it's being attacked by a nicotine-resistant caterpillar. Okay, then it produces a whole bunch of other types of compounds, many of which we had no idea what they did. And I just want to talk just briefly about a group of compounds called diterpene glycosides. These are some work done by a PhD student who's just finishing up, Sven Hellig, and he's done some beautiful analytical work characterizing these molecules that are basically unknown. There are 46 of them in Nicotiana attenuata, and they are basically produced in the chloroplast by what's called the MEP pathway and the DOX pathway to produce a basic backbone diterpene structure, and that backbone diterpene structure is depicted there. It's hydroxylated and then sent out to the plant and decorated further by enzymes that add different types of sugars to them. I'll talk about that in a little bit later. But because this is a secondary metabolic pathway, the main enzyme that's involved there, this NAGGPS that is highlighted in bold there, also has 3 copies because of that the genome triplication event. And if you silence the one that's dedicated for the production of these pathways, you can completely take out the whole biosynthetic pathway by one gene silencing step. So by silencing that particular gene, we're able to make DTG-free plants. And if you fed them to caterpillars, you can see that the caterpillars basically were able to increase their growth rate almost 5-fold when they're feeding on these DTG-free plants. So even though we had no idea that these were toxic or defensive when we looked at the structures and figured out their structures, when we silenced them and produced plants that were DTG-free, the caterpillars told us that, oh, this is really a pretty nasty defense compound. And what Sven has been able to do is to identify all the different enzymes that are involved in decorating them with sugars of various sorts, glucose and rhamnosyl, and then they are malinated in addition. And that's what generates all those 48 different structures. Now, it turns out that if you look at the poop of a caterpillar, the frass that comes out the other end of the caterpillar after it's eating leaves, Sputhy Poretti, who is a PhD student who just finished up, along with Sven and Jen-K Lee, have been discovering that there's a very interesting dynamic that's going on in the caterpillar's gut as it's trying to remove particular sugar groups from these DTGs in a way so as not to expose the toxic backbone, which is toxic to the plant also, but also not remove all of them, which produces other toxic compounds. So this is a story that is ongoing. We're still working on it, but there's this wonderful digestive duet that's occurring as the caterpillar is removing certain sugar molecules and putting them back on and putting other molecules back on to protect it and detoxify this molecule as it goes through. An example of direct defenses. Now I want to switch to indirect defenses. Now, indirect defenses are based on a concept that probably every politician knows. Now, here's the basic scenario. Here's the plant. And the plant is attacked by Manduca sexta, which is its enemy, right? Now, Manduca sexta is in turn attacked by other predators that are all depicted here. There are 6 of them right here. And they, of course, are the predators of the herbivore. Now, anyone knows that the enemy of your enemy is your friend. And that is the basis of how indirect defenses work. Indirect defenses, in contrast to the direct defenses, are signals or traits that the plant produces that help predators or parasitoids find and feed on the herbivores that are feeding on them. And that's what that indirect defense loop, how that works. Now, the way it works in Nicotiana attenuata is that when Manduca sexta begins to feed on an attenuata plant, the plant recognizes it from those FACs that are in the caterpillar spit, and it activates a series of transcription factors, and it activates the production of a beautiful volatile bouquet, like a Chanel No. 5, that's released not just from the attacked leaf, but the entire plant. And it's basically just producing the signal that includes a number of molecules, the most important of which is a sesquiterpene called trans-alpha-bergamatine, and trans-alpha-bergamatine attracts this little predator that's down here called Geochorus pallens, a little predator that lives around in the soil on the plant, and it's basically listening, smelling in the air, and when it senses that molecule, it knows there's a caterpillar feeding on a plant somewhere. But that little Geochorus also needs local information. Once it arrives on a plant, the plant is big, the caterpillar could be anywhere in the plant, and it utilizes other compounds like these green leafy volatiles in the top there, and particularly the change in a double bond in those green leafy volatiles that gives it local information and allows the Geochorus to be able to localize where on the plant that particular caterpillar is feeding. And when it gets there to the caterpillar, it just plunges its beak inside the caterpillar and sucks it out. And it does that many times. And so what this process is just like calling the police. It doesn't have to do anything more than simply provide accurate, honest information about where a caterpillar is feeding on it, how it's being attacked, and then the predators take it from there. It's a wonderful evolutionary stable way of dealing with defense because the co-evolutionary loop between plant and herbivore is broken by this predator link. Now, we discovered that thanks to the brilliance, really, of the graduate student in the group, Andre Kessler, who's now a professor at Cornell, and he invented a predation assay that allowed us to monitor the behavior of this predator in the field under natural conditions. And the predation assay was beautifully simple. He simply just glued eggs of this Manduca onto the bottom of leaves and use those eggs as a monitor for whether or not the predator had come up to the plant. The predator is a very skittish predator. It's called the big-eyed bug. It has big eyes. It pays attention to a lot of things. You can't walk up and see it. It runs away. And so you need an indirect way to know whether or not it's been around. And yet, when the predator feeds, you can see that it sucks out the egg, and it leaves the egg in a nice state behind. And by gluing eggs onto the plant, you can see how many predators have come up and visited the plant. And that predation assay had allowed us to be able to work out the transcription factors that regulate volatile production, which volatiles are important, the long and short distance signals, all the details of this particular process. Now, it turns out that these indirect defenses don't work alone. They work in synergy with the direct defenses. So when the caterpillar attacks the plant and it causes the plant to produce this wonderful volatile bouquet that is functioning as an alarm call, bringing in predators from long distances away that then attack the caterpillars, there are other things going on too, namely that the plant is also producing compounds that are interfering with the digestive process. And these are the protease inhibitors that Jorge Zavala worked on. And the protease inhibitors, here's a 7-domain protease inhibitor, and what they do is they interact with the digestive enzymes of the caterpillar's gut and keeps the caterpillar from digesting, which means that the caterpillar can eat and eat and eat, but it doesn't grow because it's not getting the nutrients. Now, when a caterpillar goes through the stages from being small to large, it becomes pretty immune to this predator because it's a bratwurst-sized caterpillar at the end, and it pretty much can thumb its nose at these little predator who's trying to attack it. But if the plant keeps the caterpillar in a nice, small, vulnerable stage longer, the indirect defense of the predator works much better. So it's the synergy between direct and indirect defenses that really helps to lower the population of caterpillars. Now, there's another type of synergy that occurs as well. And this is depicted very nicely in some videos by Mary Schumann, who is pretending to be a Geochorus predator, sticking a little blue pin up the butt of the caterpillar. And you can see on a caterpillar that's feeding on a wild-type plant that's full of defenses, it's behaving pretty sluggishly. It's not moving at all when she's poking it. She picks it up with the forceps. It doesn't do any wanging. It just hangs there limp like a doornail. Now, remember, this caterpillar is spending a lot of metabolic energy detoxifying the defenses that are in the leaves, the direct defenses. And it doesn't have a whole lot of energy to fight back when it's attacked by predators. Compare that when Mary tries to poke a caterpillar that's feeding on a protease inhibitor-free plant. It's got plenty of energy. It's banging around, it's thrashing, and it's defending itself quite well. And that's another example of the synergy between direct and indirect defenses, is that caterpillars that are feeding on toxic plants are lethargic. They are having to spend a lot of energy detoxifying all those metabolites that are going through them, and that slows them down and makes them much more vulnerable to their predators. We so frequently forget, because we eat defenseless plants in our normal food supply. We've made them defenseless through our agricultural practices, that we forget that eating native plants that are full of chemicals is actually hard, metabolically demanding work. Now, there's another type of indirect defense that I want to tell you about, and that's an indirect defense that occurs in the trichomes, which are these little hairs on the surface of the leaves, and you can see as a little droplet appearing here from this magnification of a trichome on the surface of an attenuata leaf. Now, in the trichome is a particular type of compound called an acyl sugar. Now, acyl sugars were thought to be direct defenses, toxins, and there's a good bit of evidence that they are sticky substances that catch insects and sort of tie them up. But and this was actually first worked on by Alexander Weinhold in the group, and Alexander characterized the structures of these things, and these acyl sugars basically consist of a sucrose molecule, and then on each of the hydroxy groups of the sucrose molecule is esterified a small short-chain fatty acid. Here are the characteristics of these short-chain fatty acids, and these short-chain fatty acids have the smell of baby barf. They're sort of an unpleasant smell, and that's the reason why Alexander actually started the project in the beginning, because he had to take care of the caterpillar colony, and he always thought that the caterpillars smelled fairly bad, and noticed that when they were feeding on these leaves, they were, of course, eating acyl sugars. And when we took plants to the field and noticed what caterpillars did when they first hatch out of their egg, we noticed that these acyl sugars are not defensive at all. They're, in fact, the first meal of a caterpillar. A caterpillar hatches out of its egg, and it begins to lick these tops like they're little lollipops, and they get their first meal. And in the process of getting that first meal, they end up getting a body odor. And the body odor comes from eating those acyl sugars and having those fatty acid groups deesterify and come off the body. And so the caterpillar begins to smell of those baby barf fatty acids that are esterified to those sugars. Now, we were very interested to know whether or not smelling attracted the attention of predators that were on the plants. And so we looked at all the predators that occur on plants, and none of them cared about these baby barf smells. They didn't seem to respond more to caterpillars that were scented or non-scented. So we began to think some more. But it turns out that there was another thing that these compounds did to a caterpillar's body odor. Not only did it change the body odor, but it also changed the smell of its poop. There was a caterpillar just pooping there. And poop, when it falls, usually falls according to the laws of gravity. It falls down. It doesn't always hit the fan, as the metaphor goes. The caterpillar, when it poops, produces a smelly, fresh, redolent poop that falls directly on the ground. And this is Utah, where the ground is hot. It's frequently 50 degrees, and those are short-chain fatty acids, so that they immediately volatilize. And after 5 minutes or so, they become scentless, and they no longer have that smell. But for 5 minutes, when the fresh poop has fallen on the ground, it's providing beautiful information to a whole other group of predators. And those are the predators that are walking along on the ground, the lizards and the ants. And it turns out that the lizards and the ants use that volatile information to know that, oops, there's a caterpillar above them. They can just climb up the plant. And you can take fresh frass and dried frass, or you can just isolate those fatty acids and make your own little perfume, which will be available in duty-free shops soon, and call it the scent of the caterpillar. And you can spray it on the ground and spray it on sticks in front of ant nests, and the ants will just come charging up after you've sprayed them looking for caterpillars. And so in the end, these trichomes may well be the first meal for the caterpillar, and they're delicious, sugary lollipops, but in the process of scenting their bodies and scenting their frass, they actually turn out to be evil lollipops because they tag them for predation. And that's just another example of how a plant is utilizing indirect defenses to protect themselves. They have very clever ways of bringing in predators. And that was the fourth layer. Now, I'm going to go to the fifth layer now. And the fifth layer activated by those fatty acid amino acid conjugates that are in the caterpillar spit. And the fifth layer is a layer of tolerance responses that the plant activates. We had talked earlier in part one about how a plant is a growth machine, fixing carbon dioxide, taking that carbon dioxide, making a whole bunch of metabolites for growth, reproduction, storage, and defense. But at the same time, it's also possible to use it to make the plant more tolerant of herbivore attack. Now, until this work, the whole tolerance thing was pretty much a traitless concept, something that you could look at in populations of plants, but not something you could really nail down to a particular trait. And here we've been able to nail it down to a particular trait, and it came again from a field observation. The field observation was that caterpillar attacked plants after the plants had senesced and dried out, and then there was another rain. They frequently reflowered. They produced new flowers after a rain, but the plants that were not attacked by caterpillars didn't do this reflowering. So that was an interesting observation. And you sort of wondered, where did these caterpillar attacked plants get the resources to reflower? This is an annual plant. It should have shut down life, made all the flowers that it could have, and senesced and called it quits. But that's not what they were doing. And I think the answer comes in the life history of the caterpillars that feed on them. Caterpillars go through two stages. They have the eating machine stage, which is depicted right here, when Manduca sexta is simply just the larvae trying to consume as much plant material as possible. But then it pupates and molts into this beautiful moth, and it becomes a sex machine. And as a sex machine, it's no longer eating the caterpillar and eating the plant anymore. And that means that the caterpillar is out of the concerns of the plant. And the plant, if it had waited and stored resources somewhere else, it was able to reflower and start that whole process over again without having the tissues being lost. And this is what is happening. And this is work done by Jens Schwache in his PhD project, and he discovered that the FACs in caterpillar spit, they elicit a bunkering of photoassimilates into the roots. Now, a plant is a growth machine, right? It's assimilating carbon dioxide from the air, and normally it would be fixing that carbon dioxide into sucrose and sending it from source leaves up to sink leaves to grow more leaf area to make more of a growth machine. That's what plants normally do. But if they're making more of a growth machine, they're also making more leaves for the caterpillar to eat. So you need to stop that process. And when you have a caterpillar on a plant, or you put FACs on a plant, and it doesn't matter where on the plant, instead of taking that fixed CO2 and sending it up to young sink leaves, it bunkers it down below ground. And Jens was able to show this with some beautiful experiments in collaboration with the phytosphere Jülich, which has a synchrotron, is able to make C11 carbon dioxide. C11 has a half-life of 15 minutes, so you have to be right close to the synchrotron. You can't ship it very far, and it allows you to look at very short-term partitioning of carbon in a plant after it's fixed and where it moves it around. And here's just some of the data from Jens's work. He was able to show that up is transport of C11 labeled CO2 into young leaves, and you can see that it goes, when you just wound and water a plant and you treat the wounds with water, the fixed carbon dioxide goes up the plant, but if you add spit to the wound, it goes down. It's the specific FACs in that spit that cause it to go down. And he was also able to show that there's a particular subunit of a SNRK kinase which is regulating that. This is this GAL83 subunit that is down-regulated by the FACs. And that's sort of the master sink source regulator, the genetic element that is causing this response. And that bunkering, having put that carbon down below ground into the roots, allows the plant to reflower, make bigger flowers after the caterpillar has gone. So in many ways, this level, this response, this number 5, is the kind of response that Mahatma Gandhi would have against a predator. You just sort of lay low and let it go by and don't engage in a fight, but just regrow and be able to start again. Okay, the 6th layer and the last layer is probably the most intriguing layer. It's a type of avoidance of this herbivore, and it's an avoidance response that has to deal with a fairly common natural history problem that all organisms have, and that is that some of their interactions are with good guys and some of them with bad guys, and sometimes the good guy and the bad guy are a part of the same genome. So this moth is a good guy. It's a pollinator for the plant, but it lays eggs that are bad guys that grow into little herbivores that sometimes turn into very big herbivores that are very disastrous for the plant. And the 6th response has to do with dealing with this herbivore by dealing with its mother, its pollinator. Now, I told you in session 1 that this is a plant that attracts that pollinator by producing a compound called benzyl acetone, which is depicted up there above the flower. What Danny Kessler discovered is that when the moth is attracted by that particular structure of benzyl acetone, not only is it attracted because of the nectar, it nectars and then it oviposits. So nectaring and oviposition are linked processes. The more they get nectared by and more visited by this pollinator, the more eggs show up on the plant. The eggs, of course, turn into herbivores, and therefore, the more pollination services you get, you might end up getting more herbivores if the other types of defenses I've talked about earlier aren't effective in cleaning out those herbivores and getting rid of them. Now, we were able to silence benzyl acetone production, and when we know that if the plant is not producing benzyl acetone, it's pretty much ignored in terms of pollinator activity and also oviposition activity by the moth. And Danny Kessler, who is a remarkable photographer but also a remarkable observer of natural history, noticed that attacked plants, when you looked at this, let's do it again, at this day-night transition, that the plants were beginning to produce a different type of flower after they were attacked. They were producing their normal night flowers, but then they started, when they were attacked, by producing a different type of flower that was really only opening in the morning. Now, here is the difference between the morning open flower on the bottom and the night open flower at the top. The normal flower is the night open flower, the one here. And you can see that it opens up in the first night open, and it opens and scents and attracts the moth, and then it closes a little bit for the day, and then opens again and attracts the moth again for the second night. The morning open flower stays closed that first night. It doesn't scent, and it doesn't attract any moths. And then it opens up just slightly in the next morning, and it attracts a different pollinator, and this is the pollinator, a hummingbird. And the hummingbird has the very nice characteristic that it lays hummingbird eggs, not caterpillar eggs. And by switching its sexual system to a different pollinator, asking for a different type of postman to bring gametes to you, the plant has basically solved its herbivore problem. That's pretty remarkable. So what I've done is told you about all of these changes that occur in the plant when it perceives these compounds here that are in the spit of the plant, the spit of the caterpillar as it chews on the plant, and elicits this very complex defense, avoidance, and tolerance responses. And what I've also told you, I hope, there's basically three messages behind this remarkable transition that occurs in the plants when it sees these spit factors. The first, of course, is that direct defense is not the only way of coping with herbivores. And most of our agricultural practices dealing with protecting our crop plants have to do with direct defenses. Insecticides that directly kill the crop pests. Now, as we've learned from this story, there's many other ways of dealing with your herbivore. And we should be thinking about how to incorporate some of those many other ways into our cropping systems, because some of them may well be much more evolutionary stable than just using direct defenses alone. The second main take-home message that I want you to get to is this interplay of the importance of knowing mechanism so that you can use mechanisms to be able to manipulate function. And when you can manipulate function, you can begin to ask in an unbiased way what is actually happening in nature between plants and insects and all the other interactors. And the third main message I want you to get from this is that you can observe an awful lot by just watching. Now, this little tautology is something from Yogi Berra, but I think it applies so cogently to biology today, because we don't teach our students how to watch, particularly not natural interactions anymore. This is not part of our biological training programs. And so much of the innovation that I've just shown you comes from simple natural history observations. Okay, so in the third part, that was the end of the second part. In the third part, I'm going to talk about seed, sex, and microbes. In part one, I told you that this is a plant that chases fires. It produces seeds that have to live in the seed bank for hundreds of years before the next fire comes along. And I'm going to be talking about how it uses sex to get the best genetic material to be able to survive that long time period as it waits for the next germination event, and also how it recruits microbes when it does decide to germinate in opportunistic mutualisms to help protect it against all sorts of stresses that you could hardly predict if you were been in the seed bank for hundreds of years. So I want to thank you for your attention, but I particularly want to thank both the funding organizations that make this work possible, the long-term patient funding of the Max Planck Society, and the grants we've received from these wonderful agencies that are so unbureaucratic in their administration and really promote curiosity-driven science in the best way possible. I want to thank the folks at Brigham Young University, particularly Dr. Larry Sinclair, Ken Packard, and Heriberto Madrigal, that make this wonderful interaction with that remarkable university work and allow us to use their Lytle Ranch Preserve as a laboratory to study these field interactions. And I want to thank all the people who have provided the stunning pictures and movies, the talented photographers and folks in the group who have helped support and make some of these slides, and particularly Erna Buffy and Folker Arts, who are really masters of translating science, making beautiful movies, and allowed us to use many of their outtakes from their movies in this presentation. And you for your attention.